Okay, if we can get started again. All right, so we've talked about then a little bit about the biology of phage. What does all this have to do with this process of transduction, the second gene transfer mechanism? Again, remember, transduction is gene transfer from donor to recipient by means of a bacteriophage. So how do phages then transfer genes, donor genes, to the recipient? Well, just to point out, obviously this type of gene transfer, unlike transformation, is going to be resistant to environmental nucleases, because when a phage transfers a donor gene to a recipient, the donor DNA is going to be encased in this protein coat of the phage, which protects it from the environmental nucleases. Okay. All right. Transduction. There are actually two different ways that phage can transduce genes from donors to recipient. One form of transduction is called generalized transduction. And generalized transduction is transduction in which potentially any of the donor chromosomal genes can be transferred to the recipient. All right? So it has the potential. Now, I'm not saying that all the donor genes are transferred, because even in this type of gene transfer, only a small piece of the donor chromosome is being transferred. Okay? But potentially any gene can be transferred from the donor to the recipient by these generalized transduction or generalized transducing phages. So how do they do that? All right, so here we have a donor cell. The donor chromosome is going to be in red as we go through this, and the recipient is going to be in blue. And here's our phage infecting a donor cell. All right, so there's the phage DNA. So what do these phages do? Of course, they enter the cell, and they start taking over the biosynthetic machinery of the cell, and they shift synthesis from host DNA RNA protein synthesis to phage DNA RNA protein synthesis. All right. Now, one of the things that these phages do, these generalized transducing phages, is they break down the bacterial chromosome. And the reason that they break down the bacterial chromosome is they're going to use the components, the nucleotides released from the digestion or the breakdown of the bacterial chromosome. They're going to use those as substrates to make phage DNA. And of course, the way they distinguish their DNA from the donor chromosome is that they have these modified bases in their DNA. So they digest, and they have enzymes that digest DNA, but only DNA that doesn't have these modified bases. So the donor chromosome gets broken down into a bunch of pieces, okay? And then as the infection process proceeds, you have then within the cell, you have pieces of the donor chromosome as well as a lot of phage DNA, because the phage is replicating its DNA. All right? And then eventually we begin to assemble phage particles. And these phages package the DNA in a pretty sloppy manner in a way, because there's nothing special about the phage DNA that allows it to be packaged into the phage head. These phage package DNA by what is referred to as a head-full packaging mechanism. They just stuff DNA into the head until the head is full. And they don't care what DNA it is. Well, in this cell, you've got pieces of donor DNA. So every once in a while, some of the phage particles actually get packaged with donor DNA. So here we have a phage particle that has picked up a piece of donor DNA. Now, as this infection proceeds, this cell is going to lyse, all right, and we're going to release the progeny phage. Now, if this phage particle here that has now picked up donor DNA infects another bacterial cell, which is now going to be our recipient, what we've essentially done is introduced a piece of the donor chromosome into the recipient cell. All right? Now, once this gets in here, now we're going to have, we're back in a situation like we saw with transformation. We just simply have a piece of the donor DNA in a recipient cell. So there's going to have to be a recombination event, a homologous recombination event to get stable exchange of donor and recipient genes. So homologous recombination was going to be required to get the stable integration. Now this is called generalized transduction because potentially any of the donor genes can be taken up and transferred by this process. Now, any individual phage particle will only transfer a small part of the donor chromosome. But since this is a random event, which piece of DNA gets packaged into the head, potentially any gene can be transferred by this mechanism. And that's why it's referred to as generalized transduction. Okay? The second type of transduction is referred to as specialized transduction. And this is transduction in which only certain donor genes can be transferred by the phage particle to the recipient, all right? And specialized transduction is mediated by lysogenic phages. So let's go back to our example of lambda. Here we have our lambda inserted into the chromosome. And remember I said that this is a very specific place where it's inserted at that attachment site. And in the case of lambda, they are between the genes for galactose utilization and biotin biosynthesis. Now, when the lambda lysogen or the lambda prophage is induced and gets out of this state of lysogeny, normally what happens is it goes down this pathway and pinches out, and this reverse recombination event occurs precisely as it's supposed to excise lambda from the chromosome. And that's what happens most of the time. But every once in a while, a mistake is made as this thing loops out, and the recombination event occurs at the wrong spot. And what happens as a consequence of that is that what you excise is lambda, which has now picked up some of the donor genes. And in the case of lambda, this can loop out either to pick up these galactose genes, or it can loop out in the other way to get the biotin genes, all right? But lambda could only transduce those genes that are close to where it had inserted. Either, in this case, either galactose or lambda or biotin. And in our example, it picked up galactose, okay? So, once this is out, and this now replicates itself, lambda is going to replicate itself, and it's going to package the DNA. It's going to undergo lytic growth. It's going to package the DNA. It's going to release these progeny phages that have a genome like this. If this phage now infects another recipient cell, what you've essentially done is you've transferred the donor galactose gene to the recipient. All right? Now, once this gets into the recipient, in this case, you do not necessarily require homologous recombination. All right? The reason you don't require it is because you've got lambda here that can catalyze this site-specific recombination event to get this into the recipient chromosome. So you don't have to have a reciprocal homologous recombination event. Now, it is possible that homologous recombination can also occur to exchange the donor and recipient galactose gene, but it's not necessary. Okay? Because lambda can insert itself into the chromosome anyway and enter the lysogenic state. If lambda does insert itself into the chromosome, then obviously that cell has two copies of the galactose genes, its own copy plus the copy that it received from the donor. All right? And so this is called specialized transduction because each transducing phage can only transfer in certain genes. And those genes happen to be the ones that are close to the site at which that phage inserted into the bacterial chromosome. Now, of course, different transducing phages will transduce different genes. In the case of lambda, its attachment site is between galactose and biotin. In the case of another specialized or lysogenic phage called Phe80, that one inserts close to the tryptophan genes. So that transducing phage transduces tryptophan genes. All right? So each specialized transducing phage only will transduce certain genes. Happens to be those that are close to where they inserted. All right. So what's the significance and importance of transduction? Amongst the gram-positive bacteria, this is a very common way that genes are being expressed by transducing phages, either specialized or generalized transducing phages. And another place where this plays an important role is in this phenomenon called lysogenic or phage conversion. And what this is, lysogenic or phage conversion, simply means is a change in the properties or the phenotype of a recipient cell as a consequence of being lysogenized with a lysogenic phage. So in the case of lambda we just described, what happened in the case of lambda was it now picked up the galactose genes from the donor. So it changed its properties because now it has galactose genes from the donor. But there are a number of situations where this actually results, lysogenic conversion or phage conversion actually results in increased virulence of the organism. And the classic example is in this case of diphtheria, Corynebacterium diphtheriae. Diphtheria, we haven't had those lectures yet, but diphtheria is a toxin-mediated disease, all right? And so what happens in diphtheria is that the organism, Corynebacterium diphtheriae, elicits this toxin, and the toxin mediates the disease. Well, if you look in the Corynebacterium genome, if you look in the genome of that bacterium for the toxin gene, it doesn't encode a toxin gene. So the bacterium actually, in its chromosome, does not contain a toxin gene. The only bacteria that can cause disease, or the only Corynebacterium diphtheriae that can cause disease, are those that have been lysogenized with the phage, because there is a lysogenic phage that actually carries the toxin gene. And so those bacteria that harbor that prophage that carries the toxin gene are pathogenic. Corynebacterium that do not harbor the prophage are not pathogenic at all because they can't make the toxin, okay? So this is an example where phage conversion or lysogenic conversion results in virulence of the organism. So in a sense, you can say that the phage is causing this disease. Now I don't want you to, please don't say that I told you that Corynebacterium diphtheriae does not cause diphtheria. It does. But it does so with the help of the phage, because the phage can't make the protein itself. They rely on the bacteria to make that protein. Okay? But phage conversion occurs. And there are numerous examples of virulence genes being actually carried by phage that lysogenize bacteria and result in disease or increased virulence. All right. The third transfer mechanism is referred to as conjugation. And conjugation is gene transfer from a donor to a recipient by direct physical contact between the donor and recipient cells. This is essentially sex in bacteria. Okay? In bacteria, there are two mating types. They're generally simply referred to as donor and recipient, although sometimes you do see them referred to as male and female strains of bacteria, but more commonly it's referred to as donor and recipient. And a donor is a donor because it harbors an extra piece of DNA, which is referred to as the F factor or fertility factor. Now, this F factor is actually an extra chromosomal piece of DNA, and it's an example of a plasmid. Plasmids are extrachromosomal pieces of DNA. So this is an example of a plasmid. This is the F plasmid. Now, F is the first, the F factor or the fertility factor was the first of these factors to be described. There are actually many plasmids that mediate the same process. But we're going to use the F factor as our example of how conjugation works. But keep in mind, there are other plasmids that do the same or similar things. So donors are donors because they harbor this extra piece of DNA called the F factor. And this F factor has a number of genes on it, one of which codes for a very unique pilus that is expressed on the surface of these donor cells, which is referred to as the F pilus or the sex pilus. All right? And this is different from the pili that Dr. Fox talked. This is a very unique pilus that is involved only in this conjugation process. The recipient cells, the other mating type in bacteria, are recipients simply because they lack an F factor. So donors are donors because they contain this plasmid, this F plasmid. Recipients are recipients because they lack it. All right, now that F factor can actually exist within the donor cells in three different forms. And what we want to do is to talk about the three different ways that F factor can exist in the donor cell. Describe the characteristics of crosses between donors and recipients when F exists in that state. And then we're going to go back and talk about the mechanism and see how the mechanisms of conjugation explain these various characteristics. So the first way the F factor can exist is autonomously. As an extra chromosomal piece of DNA. In the cells, donor cells harboring an F factor in this state are referred to as F plus cells. And so if you have a cross between an F plus cell and an F minus recipient, what will happen is after the conjugation event has occurred, the F minus, the recipient in the cell, will be converted into a donor. So the F minus, the recipient, will become a donor. The donor, the original donor, will remain as a donor. And there will be a low frequency of transfer of donor chromosomal genes. So we're going to convert the recipient to a donor, we're going to keep the donor a donor, and we're going to have very low transfer of these donor genes on the chromosome. All right? The second way that F can exist is actually integrated into the chromosome. Cells harboring an F factor integrated into the chromosomes are referred to as HFR strains. HFR stands for high frequency of recombination. All right? And you'll see why it's referred to that in a second. All right, so how does F get into the chromosome? Well, it gets in by a reciprocal recombination event. Again, there we have two circles. A single recombination event between two circles results in integration, just as we saw with lambda. So the F factor can actually integrate into the chromosome by a recombination event. All right, so we have now an HFR strain. So in crosses of the type of an HFR with the recipient, what happens is that in this case, the recipient remains a recipient. I'd say it rarely becomes an HFR. This was the exception to the rule when I first started talking about gene transfer. In the laboratory, if you take extreme care so that there are no shearing forces whatsoever that can disrupt the mating pairs, you can, in fact, get the recipient converted into an HFR strain. But in nature, that probably never happens because there's always shear forces that disrupt the mating pairs, and you never get conversion of the F minus into an F plus, and you'll see why in a second. Okay, so in the real world, it never happens. Okay, in the laboratory, we can make it happen, but for our purposes here, since we're talking about real world things, the F minus is not going to become an HFR. It's going to remain an F minus. The HFR, however, will remain an HFR. And if you look then, there's going to be a high frequency of transfer of donor chromosomal genes. That's why these are called high frequency recombination strains, because there's a high frequency of transfer of donor genes, and there's going to be recombination required in order to get exchange, as we'll see shortly. The third way that the F factor can exist is again in an autonomous state, but now carrying some extra donor genes. Donor cells harboring an F factor, in this state I refer to as F prime cells. And sometimes you see after the prime, there'll be some other designations. And those other designations essentially tell you what genes it's carrying. So for example, if it said F prime lac, that would be an F prime factor that is carrying the lactose genes. Okay? So what follows the prime is usually some designation telling you what donor genes are being carried. All right. So how do you produce these things? Well, again, just like we saw in lambda, if the F factor can insert into the chromosome, it can also excise from the chromosome. And while in most cases it excises correctly, every once in a while a mistake is made when you excise the F factor so that you pick up some of the donor genes. And again, the genes that can be picked up are genes on either side of where it has inserted into the donor chromosome. So here we have an F factor that has now picked up some donor genes, okay, and now exists autonomously. So this would be called an F prime factor. So in crosses of the type F prime by F minus, in this case the F minus will become an F prime, the F prime will remain an F prime, and there's going to be a high frequency of transfer of those donor genes that are carried by the F prime factor, but low frequency of transfer of other donor chromosomal genes. Okay? All right, so now let's go look at what happens mechanistically, what happens in this process, and see how we can explain these various characteristics. And we'll start by the F plus by F minus crosses. All right, the first step in conjugation is pair formation. That F pilus makes contact with the recipient cell, and a conjugation bridge is actually formed between the two cells so that we have a continuum of the cytoplasm of these two cells. All right, so that's referred to as pair formation, or the conjugation bridge is formed. And that is that F pilus that forms that conjugation bridge. The next step is the DNA transfer event. But this is a very unique type of DNA transfer and type of replication. It's referred to as rolling circle replication. What happens? There is a very unique site on the F factor. Here, I don't know if you can see it there, it's indicated by that black dot. That is called the origin of transfer. And what happens in this gene transfer mechanism is that a nick is made in the DNA at that point. But the nick is only made in one strand of the DNA molecule. So you have one nick strand and one unnicked strand in that DNA. Now, once you have the nick there, you have a free 3' hydroxyl group on one side, right? You remember back from your biochemistry. And what do DNA polymerases do? They add nucleotides onto a primer, which is has a free 3' hydroxyl group, and they read a template in order to determine the correct sequence. Okay, so you have a 3' hydroxyl group, and the unnicked circle is going to act as the template, and so you're going to replicate this DNA, this piece of DNA, but as you're doing that, of course, you're displacing one of the strands of the DNA. All right? And so what happens, and this is called rolling circles, you envision the circle just sort of rolling around, spinning out and displacing one strand of DNA as it's being synthesized. So the donor is synthesizing the new DNA and displacing one of the strands. That strand makes its way through the conjugation bridge into the recipient. And in the recipient, the reciprocal strand is then made. All right? Now, eventually, shearing forces in the environment are going to disrupt these mating pairs, and you're going to have these things separated, okay? But now notice what has happened. As the consequence of this conjugation event, the original recipient strain has now been converted to an F+ strain because it's received the F factor by this rolling circle type of replication and gene transfer. The original donor has stayed a donor because even though it's giving some of the DNA to the recipient, it's replicating the DNA as it's doing that transfer, so it keeps its own copy, so it stays as an F+ cell. And notice there's very low transfer of these donor chromosomal genes. In fact, the way we've described it, there's no transfer of these donor chromosomal genes. In fact, if you look, you can, in fact, demonstrate some of these donor chromosomal genes can get transferred into the recipient. All right? It's at a very low frequency, however. And there's two explanations as to how that can happen. One explanation is that when the donor starts this unique type of replication, this rolling circle replication, in nicking the strand, that occasionally a mistake is made and you also nick the donor chromosome. And if you nick that donor chromosome in just one strand, then there's no reason why it cannot also be transferred by this rolling circle replication. And so that's one of the reasons why you get some transfer of donor chromosomal genes by this process. But it's a rare event because it requires an accidental nick into the donor chromosome. But it happens at some low frequency. The other reason, the other explanation as to why you do see some transfer in these types of situations is that the transfer may not, in fact, be mediated by the F+ strain, but it may be mediated by HFR strains. Because remember, F can integrate into the chromosome. So if you have a large population of F+ cells, there may be somewhere in that population one cell in which the F factor has, in fact, integrated into the chromosome. And that cell, as we're going to see shortly, actually transfers donor chromosomal genes quite effectively. All right? So either because some mistake is made when you nick the chromosome, or because in the population of F+ cells there may be a rare HFR strain that's doing the transfer, you can, in fact, detect some low degree of transfer of donor genes into this original recipient cell. All right, so let's look at the second type of conjugation, HFR by F- conjugation. All right, in this type of conjugation, again, the first step is the formation of this conjugation bridge, mediated by that F pilus. And again, you get DNA transfer by this rolling circle type replication process. But now what's happened? F has integrated into the chromosome. And so now when you nick at this point here, at this origin of transfer, and you start replicating this circle, what gets displaced is the donor chromosomal strand. And so the thing that actually gets transferred into the recipient is a piece of the donor chromosome, not the F factor. In fact, the only way the F factor could actually be transferred into the recipient is for you to first transfer the entire donor chromosome. And again, that doesn't happen because shear forces will disrupt the mating before the entire chromosome can be transferred. Now again, in the lab, if you take extra special precautions to avoid any shear stresses, you could get this to happen. But that's not the real world situation. In the real world, this never transfers the whole chromosome. So only a piece of the donor chromosome gets into this recipient. And the only thing that gets in is the piece of the donor chromosome. The F factor never gets in. So when although it's single-stranded, in the recipient, the complementary strand is made, and you end up then with a situation where you have a recipient chromosome with a piece of donor chromosome. Now we have to get homologous recombination in order to get stable exchange of genetic information. So in this type of conjugation, those rec genes are also going to be required because this requires homology. So we get homologous recombination event occurring to get stable exchange. All right? So look what's happened here. The original recipient has remained a recipient. The original donor has remained as a donor because, again, it's replicating its DNA as it's transferring it into the recipient. And there's a high frequency of transfer of donor chromosomal genes because it's only the donor chromosome that gets transferred. But again, notice it's not the whole chromosome, it's only a piece of the donor chromosome that gets transferred. Okay? So, which genes any particular HFR can transfer will, of course, depend on where F is inserted into the chromosome. If F inserts here, then the first genes that are going to be transferred are here. If F inserts down here, then these genes can be transferred. All right? So depending upon where F inserts into the chromosome will depend on which genes can get transferred. But in all cases, it's the donor genes that are going to be transferred. Now, notice also in this case, the number of donor genes that can be transferred can vary depending upon when this disruption of the mating pairs occurs. If it occurs very early, only a few donor genes can be transferred. If the disruption of the mating pairs occurs late, you might get a larger portion of the donor chromosome being transferred. So by this type of transfer, you can actually transfer larger pieces of the donor chromosome than you could transfer, for example, by transduction. What limits the amount of genes that can be transferred by transduction is, of course, the size of that phage head. You can only put a certain amount of DNA into that phage head. Here, you're not constrained by that. You could transfer much larger pieces of the donor chromosome depending upon what shear forces are present in the environment. Okay? All right. So here's a cartoon of HFR. You get the conjugation bridge. There's the rolling transfer. We're transferring in the donor chromosome, break the mating pairs, homologous recombination to get stable exchange of the DNA. Okay? And now we've got the F factor with some DNA. All right. The next type of cross, F prime by F minus crosses. Again, conjugation bridge formation, DNA transfer, again, the same mechanism, rolling circle replication, but now we've got the F factor is autonomous again. And so it gets transferred into the recipient. Since this is a small piece of DNA, relatively speaking, compared to the chromosome, obviously it gets transferred pretty rapidly, and eventually the mating pairs are disrupted. But now we see what was the original F minus has now become an F plus because it's inherited the F factor. The original strain remains an F plus, and there's high frequency of transfer of those donor genes that are on the F factor, and again, low frequency of transfer of other donor chromosomal genes. Okay? All right, so what is the importance of conjugation? Amongst the gram-negative bacteria, this is a very, very common way to transfer genes by conjugation. In fact, the biggest problem is, of course, is transferring of antibiotic resistance. There are plasmids out there that carry antibiotic resistance genes. And these things get transferred very, very efficiently, particularly among the gram-negatives, by this process of conjugation. And I didn't point out to it earlier, but I think it should be obvious. When you're dealing, if you have an antibiotic resistance gene, for example, so you're dealing with an analogous situation like an F prime that is now carrying an antibiotic resistance gene, that type of conjugation, is an infectious process. Every time a recipient conjugates with a donor strain, the recipient becomes a donor. And so where you start out even a single donor gene that carries an antibiotic resistant gene, after one round of conjugation, there are now two donor strains, each of which can conjugate with another recipient, and you jump up to 4 and 8, and it goes up exponentially. So these antibiotic resistance genes, if they make their way onto a plasmid, can be very, very rapidly spread through the population of bacteria. And that's what's happening in the gram-negative bacteria. We have these antibiotic resistance genes on various plasmids, and they're being distributed amongst the bacteria populations very, very rapidly. And again, I want to remind you, while this conjugation occurs most efficiently between members of the same species, it's not uncommon to also see it go across species. So an antibiotic resistance gene that's in the E. coli could get over to Shigella, which is a much more virulent organism, very easily by conjugation. Okay? Now, amongst the gram-negative, or gram-positives, I don't want to leave you the impression that conjugation does not occur. It does occur. It's not as common as you see in the gram-negatives. And the mechanisms are slightly different than the way we've described it. In the case of the gram-positives, the donor strains produce some sort of adhesive material, and you actually get a fusion of the cells. So it's not exactly through the formation of the conjugation bridge. So it does occur in gram-positive. It's not as common in the gram-positives as it is in the gram-negatives. Okay. Any questions on gene transfer? All right. So what we've seen so far is mutations can occur, and they can arise spontaneously. They may be antibiotic resistance mutations. They may be mutations to increase virulence. We've talked about then the ways that bacteria, once these things occur, how they can be distributed amongst the population of bacteria. What we want to talk about next is make even more problems, the existence of transposable genetic elements in bacteria. And so what are transposable genetic elements? These are pieces of DNA that are capable of moving around from one location to another. They always have to be in some other piece of DNA, but they actually can move from one location on the DNA molecule to another location. They may move from one piece of DNA to another one. So, for example, from a chromosome onto a plasmid, or from a plasmid onto a chromosome, or from the chromosome onto a phage DNA. They have this ability to jump around. These are essentially jumping genes. All right? They have some unique properties. They're capable of random movement. And you notice I put random in quotes. It's not 100% random, but they can, in fact, insert in many, many places. And so for practically speaking, it's almost random. So I put that sort of in quotes. Now, these elements are not capable of self-replication. They're not an independent replicon. A replicon is something that is capable of replicating itself. So these can't replicate themselves. They always then must parasitize. They must exist in some other piece of DNA. Now, these elements also have a site-specific recombination event. When they move around, they do so by recombination, but it's not by homologous recombination, so they don't use those REC genes. They have their own site-specific recombination system, which is referred to as a transposase. Now, when they do transpose and move from one location to another, they sometimes copy themselves and keep so that they remain at the original location and also get put into another location. Now, it seems like there's a sort of a contradiction. I just said that they don't replicate. And the reason I make the distinction is replication refers to copying yourself to pass it on to daughter cells. That doesn't happen with these things. But they can make a copy of themselves to pass it on to another piece of DNA within the same cell. So it's not, strictly speaking, recombination. All right? It is a duplication, but it's not for recombination. It's not replication purposes. All right? Okay, so what are these elements? The first ones are referred to as insertion sequences, or IS sequences, and these are transposable genetic elements that carry no other genes except those that are required for transposition. So they have a transposase, but they have nothing else besides that. And you'll see them as far as the nomenclature. They're all going to be called IS followed by some number. And I don't even know what we're up to now. It's IS 20 or 30. There's a number of these things that have been described, okay? So the number just referred to which one they're talking about. They have a unique structure. They have, as I said, this transposase activity, so they have some sort of genes for this transposase. And then flanking the transposase, they have some sort of an inverted, either a direct repeat or an inverted repeated sequence. So in this case, we have an inverted repeat. But it also could be, this could be on this side A, B, C, D, E, F, G as well. But it's some sort of repeated sequence flanking the transposase, either a direct or inverted repeat. And we're not going to talk about the mechanism of transposition, but just suffice it to say that this inverted repeat is this repeated sequence, either direct or inverted, is required for the transposition. If you get rid of any one of those, it cannot transpose itself anymore. So that's important for transposition. So why are these things important? Well, these things, insertion sequences, are actually one of the sources of mutations in bacteria. Because these things have the ability to jump around and insert basically relatively randomly. If they insert into a gene, they inactivate the gene. And so this is one of the ways that genes are being inactivated in bacteria by insertion sequences jumping around. These also play a role in plasmid insertion. When we were talking about F, I said that F could insert in different places on the chromosome. Actually, what happens is F tends to insert at or near the location of insertion sequences. So if there's an insertion sequence at a particular spot in the chromosome, F can insert at that place. But since insertion sequences can jump around and move around any place, theoretically, F can also insert any place. So they're involved in plasmid insertion. But most importantly, they're also involved in phase variation. Again, phase variation is this change in the antigenic properties of a cell as a consequence, in this case, of an insertion sequence. And the classic example of that is phase variation in the Salmonella H antigens. Now, there are a number of antigens present on bacterial cells that are important to which we mount an immune response. The O antigen, Dr. Fox already mentioned to you, that's the lipopolysaccharide. The carbohydrate portion of the lipopolysaccharide is referred to as the O antigen, and we mount an immune response against that. Another type of antigen is called the H antigens, and these are the flagellar antigens. So they are also important. We mount immune responses against the flagellar antigens. So in the case of Salmonella, there's phase variation in the flagellar or the H antigens. And again, this is a way for some Salmonella to avoid the immune response. So if we get infected with Salmonella with one type of flagella, we start making an immune response against it. These flagella switch their phase and start making another antigenic type, to which we then have to start all over making antibodies. And in the case of Salmonella, this phase variation occurs because of an insertion sequence. And the way that happens is that there are two genes for the two antigenically different forms of the flagella, the H1 gene and the H2 gene. These genes are, between the two genes is the controlling region, is the promoter for these two genes. And this promoter is actually carried by this insertion sequence. And what happens is if the insertion sequence is in one orientation, then one gene is turned on. So in this example, the insertion sequence is oriented in such a way that the promoter drives the expression of the H2 gene. And so this Salmonella expresses the H2 antigen on its surface. We get infected with that bacterium. We start making antibodies against the H2 flagella. And those would be protective antibodies. So what happens? This insertion sequence has the ability to jump around and move around. And so it changes its orientation and is oriented in this way. So that now the promoter drives the expression of the H1 gene. So while we're mounting an immune response against the H2 antigens, the Salmonella switch over to make H1 antigenic type flagella, evade the immune system, at least temporarily. Eventually, of course, we'll start making antibodies against this. But for some time now, it's going to be about a week, roughly a week to 10 days, the Salmonella has an advantage because it has changed its phase. So phase variation in the flagellar antigens in Salmonella is actually mediated by an insertion sequence. Okay, the other type of transposable genetic element is called a transposon, or TN. And again, these are transposable genetic elements that carry other genes besides the transposase. So the insertion sequence, basically these are functional definitions. The insertion sequence has no other genes except the transposase. Transposons carry other genes. All right, and again, the nomenclature is just TN followed by some number. And of course, the genes that concern us in this class that these things carry very often are antibiotic resistance genes. Okay, and structurally, they have, again, the same type of thing. They have I haven't shown here the transposase, but they'll have the transposase, but then they have one or more antibiotic resistance genes located in between here. And again, flanking those things are these repeated sequences. And they may be a direct repeat or an inverted repeat, just like we saw in the insertion sequence. In fact, sometimes the repeated sequences on the ends of these things are actually insertion sequences. So you'll have an insertion sequence here and the same one inserted here, either in a direct way or in the reverse way. Again, and those are important for the transposition event to occur. Transposons that are composed of have insertion sequences as their repeated sequences at the ends are referred to as composite, because they're made up of, it's a transposon and insertion sequences as well. And of course, the importance of this is, as I said, is they carry antibiotic resistance genes. All right, and the reason that these are important, remember, these things are transposable genetic elements. So once you have a antibiotic resistance gene on a transposon, it can very easily be transferred to another piece of DNA. So let's say you have a bacteria that has a transposon carrying an antibiotic resistance gene on its chromosome. That bacteria conjugates, it's a recipient strain, it conjugates with the donor strain and now receives an F plasmid. Nothing can stop that transposon then from jumping from the chromosome onto the plasmid. Once it's on the plasmid, now it can be very efficiently transferred by conjugation to all the cells. So you very rapidly spread that resistance marker throughout the population. And similarly, if it were on the chromosome, there's nothing to stop it if that bacterial cell gets infected by a phage. It can jump onto the phage DNA, and then the phage can spread the antibiotic resistance gene around to the other bacterial cells. So once these things make their way on transposons, they can be very rapidly spread, and that's why we have problems with antibiotic resistance. All right, just a couple comments on plasmids. Again, these are nothing else but extrachromosomal pieces of DNA that are capable of self-replication, okay? And F is an example of a plasmid. Now, you'll sometimes see this term used, episome. And let me just tell you what it is. I don't tend to use that term, so I'll talk about plasmids. And nowadays, most people are switching over and just referring to plasmids. But there was an older term that was used, the term episome. It is actually refers to plasmids that can be shown to integrate into the chromosome. So if a plasmid, like F is an example, that it can be shown to integrate into the chromosome, it's referred to as an episome. So F is an example of an episome. If a plasmid could not be demonstrated to actually insert itself into the chromosome, then it was referred to as a plasmid. As I said, the distinction between these nowadays is going away. Most people just refer to these things as plasmids. Whether or not they can integrate, it doesn't make any difference. And that's the way I'm going to do it. I'll just refer to them generally as plasmids. The plasmids are classified in a number of different ways. One is based on the transfer properties. Okay, there are conjugative plasmids and non-conjugative plasmids. Okay, obviously these mediate conjugation; these do not. But I should tell you that even though non-conjugative plasmids cannot conjugate and mediate conjugation by themselves, that doesn't mean that they're not of concern. Because if a non-conjugative plasmid finds itself in the same cell as a conjugative plasmid, the conjugative plasmid actually can mediate transfer of both. So even an antibiotic resistance gene that may be found on a non-conjugative plasmid, while it can't be transferred if that is the only thing the bacterial cell has, if that cell also then gets a conjugative plasmid by conjugation, that conjugative plasmid will transfer both plasmids by conjugation. So even an antibiotic-resistant gene on a non-conjugative plasmid can ultimately be transferred to other bacteria cells as well. They're also classified, plasmids can be classified on their phenotypic effects. The original, the F factor, was classified based on its fertility. There are some plasmids that are referred to as bacteriocynogenic plasmids. These are plasmids that make colicins. And colicins are proteins that kill other bacteria. Bacteria use these basically as a way of competing with each other. So if you're a bacterium and you're competing for foodstuffs with some other bacterium, make a colicin that kills the other bacteria. And these are plasmid mediated. But the plasmids that we're really interested in are these R plasmids or resistance plasmids. And R plasmids carry antibiotic resistance genes, obviously. And these are conjugative plasmids. All right? And they actually structurally, they have a portion of the molecule which is referred to as the RTF, or resistance transfer factor. This is the part of the plasmid that contains all the genes that are necessary for conjugation. So this would code for that F pilus, for example, and anything else that is needed for conjugation to occur. So these are conjugative plasmids. The other part of the molecule is referred to as the R determinant. And this is where the antibiotic resistance genes are located. And in these R plasmids, or these R factors, many of these resistance markers are actually carried by transposons. So are actually found on transposons. And the reason these things are such a problem is because they're always changing. And the reason that they're always changing is because these things are on, these resistance markers are on transposons. So what happens? You have a patient that comes in with a bacterial infection. And you find out that those bacteria are resistant to, say, to the aminoglycosides and to tetracycline. All right, so you choose another antibiotic, and you start treating with another antibiotic. Well, those cells, then now when you put that selective pressure on with another antibiotic, any cell that has a resistance marker for this new antibiotic, let's say it's erythromycin, that you start treating with, you're putting that selective pressure on. So if you have an R plasmid that then passes through a cell that has a resistance factor on a transposon that would confer resistance to erythromycin, nothing can stop you for that, since it's on a transposon, jumping onto that R plasmid. So now we added an extra resistance marker. So now those cells will be resistant to erythromycin as well. And so now you try another antibiotic. And as this plasmid is being passed around by conjugation throughout the bacterial population, it picks up yet another resistance marker on a transposon. When you stop using a particular antibiotic, there's no selective pressure for that antibiotic resistance marker to stay there, so they disappear. It jumps off of this R plasmid. And so these things are constantly changing. As we use the antibiotic, we provide the selective pressure for getting a plasmid that now has picked up a new marker. When we stop using the antibiotic, the selective pressure is gone, and that antibiotic resistance marker jumps off. And so that's why these things are changing all the time, because they're on these transposons that are constantly jumping on and off these R plasmids. So as we, depending upon which antibiotics are being used at the time, that will influence what type of R factors are circulating at that time. And that's why you have such a problem with antibiotic resistance. Okay? Any questions? All right, tomorrow we'll pick up and continue talking about some genetics, where we'll talk about the gene regulation in bacteria.